So hello students of second semester Botany General. Today I'll be discussing the topic Pteris sp from pteridophytes. So first we'll come to the morphology. Pteris species, it belongs to the division Filicophyta. So the main sporophytic plant body is differentiated into root, stem, rhizome and leaves. So the habit of this Pteris species, it is a creeping rhizome which is branched, short and it is covered with scales. The roots, they arise from the lower surface of the rhizome and the leaves are pinnately compound, petioles covered with scales, pinna sessile, which means that the leaflet, each leaflet, it does not have a stalk and it tapers towards the apex. So the habitat, it is found in moist and shaded places and about 250 species are known all over the world. So a pinnately compound leaf looks like this. There is a long rachis, okay. It's not exact, but I'm just showing what a pinnately compound leaf looks like. Okay. This is a pinnately compound leaf. This is the rachis. This is the leaflet. You can also call it as a pinna. And this is the petiole. They say that the pinna is sessile. Pinna is sessile means you can see that the pinna is directly attached to the rachis. There is no stalk. In most of the leaves, the leaf, if this is the leaf, there is a stalk present and that is attached to the stem. So if this stalk is absent, then it is known as a sessile pinna. So if you have to see the root, the primary root is ephemeral. What is ephemeral? It is that root which lasts for a very short time, and later this root is again replaced by adventitious roots. The roots, they are small and branched. So the transverse section of a root shows an outer piliferous layer. So what is a piliferous layer? It is the newest layer of the outer skin of the root. Okay, so it is that part of the root from where the root hairs, they grow. It consists of a cortex and a central stele. Now the cortex is divided into an outer parenchymatous cortex and an inner sclerenchymatous cortex. The stele is protostelic with diarch and exact xylem. So protostelic means it is a simple stele which consists of a central core of xylem. Okay, there is a central core of xylem in this and which is surrounded by a cylinder phloem, and there is no pith. Now the rhizome. The rhizome or stem is differentiated into nodes and internodes. So rhizome is basically, it is an underground modified stem. So it is differentiated into nodes and internodes. So the entire surface of the rhizome, it is covered with scales. So the growing point of the rhizome is covered with ramenta. So from the growing point of rhizome is covered with ramenta. So the rhizome has an outer cuticle. Like you can see in the picture, below the cuticle is a single layered epidermis, a few layered sclerenchymatous hypodermis, a broad parenchymatous cortex, single layered endodermis, single layered pericycle, phloem, xylem, which is of mesoarch type. Mesoarch type of xylem means that the older formed xylem is surrounded by later formed xylem, which we can also be said in another way, like the old xylem is surrounded by new xylem and a pith is present. Petiole. So petiole is a semicircular structure. So the epidermis is surrounded by cuticle, above which multicellular hair is present called ramenta. So you can see in the picture, therefore the outermost layer is the cuticle. Below the cuticle lies the epidermis, and the ramenta, the hair-like structure, it arises from the epidermis. So below the epidermis is again sclerenchymatous hypodermis, then cortex, endodermis, pericycle, phloem, and an exact type of xylem. So exact type of xylem means the protoxylem is present towards the periphery and the metaxylem is present in the center. So this type of xylem is known as exact type of xylem. The vascular bundle is C-shaped. You can see in the center the vascular bundle, it forms a C-shaped structure. The leaf. So the leaves are borne on the upper surface of the rhizome. It is spirally coiled and they show succinate venation when they are young. So unipinnately or multipinnately compound or decomposed with a long rachis. I already showed you what is a pinnately compound leaf. Pinnae, that's small near the base and towards the apex and large in the middle. So all leaves, they are fertile, bearing sori along the ventral lower surface of the margin. The rachis is traversed by a single C-U-V shaped leaf trace. So leaf trace is the vascular supply that goes from the rhizome or the stem to the leaf. So a transverse section of the leaf shows a succinate, it shows a cuticle. So the transverse section of a leaf shows a cuticle, upper epidermis, mesophyll cells, which is divided into palisade and spongy parenchyma, endodermis, pericycle, phloem, and xylem. Presence of large pinna are present. Okay, so it is tapering. This is the meaning of tapering, and this is the rachis. Okay, and on the ventral surface, what is present? Sori are present. So if this is the lower surface of the leaf, we have the midrib, we have the lateral veins, and in the at the sides, what is present? Sori are present. Okay. Sori is present on the ventral surface of the leaf. And the leaves, they show succinate venation. Supposing this is the rhizome, okay, we have this is the rhizome, which is creeping type. There are scales present, and we have the rhizoids coming down, rhizoids. Okay, this is the, and the young leaves, they show succinate venation, means they show this kind of structure. This is known as succinate venation. Now we'll come to the reproduction. Reproduction, it takes place by means of spores. So the spores, they are homosporous. Homosporous meaning all the spores, they are of the same type. Okay, it is of the same type. And the sorus is cenosorus. Cenosorus means there is a continuous line of sporangia present along the margin of the ventral surface of the leaf. You can see in the picture, that is the ventral surface of the leaves, and there is a continuous line of sori present. So this condition is known as cenosorus. So the cenosori are protected by a folded margin, also known as false indusium of the pinnae. So you can see in the second picture, towards the edge there is a folded margin, and this is known as false indusium. Development is leptosporangiate type. Leptosporangiate type means the sporangium, it develops from a single sporangial initial, like here there is only one cell, and from this the sporangium it develops. That is leptosporangiate type of development. So first you can see the sporangial initial, it divides into upper cell and lower cell, and then if there is apical cell is formed. Okay, apical cell is formed, and from the apical cell there is a jacket initial and the archisporial initial, and then we have the stalk. So the jacket initial, it gives rise to the jacket layer, which consists of three types of cells: the annulus, the stomium, and the parenchyma cells. And this archisporial cell, it gives rise to the tapetum and the sporogenous mother cell. Okay, this sporogenous mother cell in the middle, this one here in the middle, this one, it undergoes meiosis division, which gives rise to, this is 2n. It undergoes a meiosis division and it gives rise to haploid spores. Okay, it is haploid. This is the Naturesporangia. So you can see it has a long stalk that terminates into a capsule, and the jacket of the capsule consists of 3 different types of cells: a thick-walled vertical annulus, a thin-walled radially arranged stomium, and large parenchymatous cells with undulated walls. So when the sporangium it matures, what happens is, from the stomium region, here stomium region, what happens, this it breaks and the spores they come out, and it looks like somewhat like this. Okay, from here it breaks and the spores they come out. This annulus is here. Okay, this is what it looks like. The spores they come out and they land on a suitable substratum. After falling on a suitable substratum, they germinate, and initially the spore wall, the exine, it ruptures and the inner contents come out in the form of a germ tube, and subsequently by a transverse division in the germ tube, it forms the first prothallal cell. The prothallal cell then divides to form a small filament having an apical terminal cell with 2 cutting faces. The prothallus is monoecious, protandrous. Antheridia appear first and are confined to the basal, central or lateral regions among rhizoids. Archegonia develop near the apical nodes. So you can see the gametophyte, it is a heart-shaped structure, and you can see the antheridia, they appear first and are confined to the basal, central or lateral regions among rhizoids. So the antheridia, they are confined to the basal region. In Archegonia, they develop near the apical notch. Okay, there is a small hole-like structure, that is the apical notch, and the Archegonia, they develop near the apical notch. So the spores of Pteris species, it is a triangular in shape. Okay, so it consists of this is the intine, and it consists of a sculptured exine. Okay, it is sculptured. So what happens from this, the germ tubes, it comes out. Okay, the exine breaks and the germ tube it comes out, and later on this germ tube it develops into cylindrical thallus cells. It develops into cylindrical thallus cells here, and a rhizoidal initial, and this is the rhizoidal initial. Develops from an antheridial initial, and the division continues, and at the what happens from the antheridial initial, what forms an outer jacket layer, like here you can see the jacket layer, and in the center what is formed, antherozoids are formed. Okay, so what antherozoids look like, it is ciliated, it contains a lot of cilia. Okay, it contains cilia which allows it to swim and fertilize the egg of the Archegonia. So this is what the antheridium looks like. Archegonium of Pteris, it consists of 5 to 6 cell projecting curved neck or neck canal cell and a ventral canal cell. Here you can see there is this central egg, then we have the ventral canal cell here, then a neck canal cell. This is the structure of a mature Archegonium. Once they mature, what happens? Fertilization takes place. So the antheridium at mature, when it becomes mature, it absorbs water and it swells. So due to the increase in pressure within the antheridium, the cover cells they split apart, releasing the antherozoids in a thin film of water present on the surface. So the antherozoids, they are released in a thin film of water. So because of that, they have to swim in the water. That is why the antherozoids, they are ciliated. They consist of cilia. Now what happens is, at the same time, the ventral canal cell, the neck canal cell and the neck cells at the top disintegrate, forming an open passage. Now the Archegonia, okay, you have seen the neck canal cell, ventral canal cell and the neck cells, they disintegrate. They disintegrate and they form an open passage for the antherozoids to come towards the egg, and eventually one antherozoid, it fertilizes the egg to form the zygote. The zygote is a diploid structure. Fertilization of the antherozoids and the egg, it takes place, what is formed? A zygote is formed. So, the zygote is a diploid structure. The first division of the zygote is vertical. So, vertical division, it undergoes a vertical division. Then there is this transverse division. First we have the vertical division, then later second division is the transverse division, where a quadrant structure is formed, and later on it undergoes repeated division and it reaches a 32 celled stage. Okay, and here from this 32 celled embryo the differentiation begins. Okay, differentiation begins. So, no suspensor is formed. The hypobasal cells form stem, apex and foot, while the epibasal cells forms the cotyledon and root. So, no suspensor is formed here. So, what is a suspensor? Suspensor is nothing but it helps in the absorption of nutrients for the development of the young embryo. So, then once the 32 cell stage is reached, the differentiation starts. What happens is the hypobasal cell, it forms the stem apex. Now, what is a hypobasal cell and what is an epibasal cell? Supposing this is the archegonium and we have the, here we have the zygote. So, the upper portion of this zygote is known as the epibasal and the lower portion is known as the hypobasal. So, the lower portion, it forms the stem, apex and the foot, while the upper epibasal cells, it forms the cotyledon and the root. So, with the development of embryo, the venter of the archegonium forms a protective layer called calyptra around the embryo. So, what happens is the venter of the archegonium, it forms a protective layer of the embryo. It protects the embryo, which is known as the calyptra. Now, in the young embryo, the root and cotyledon grow more rapidly than the shoot. The root pierces, now root and cotyledon, they grow rapidly, and what happens once the root grows, it pierces the prothallus, means it comes out of the gametophyte and it establishes the sporulum in the soil. Now, it pierces the soil. Later, the first leaf develops and gradually sporophyte is formed and the gametophyte disintegrates. Now, once the root it establishes in the soil, what happens? A new sporophyte develops and the gametophyte it disintegrates. So, what happens? Now, the sporophytic generation again it begins. So, if you have to see the life cycle, first we see the sporophytic plant that we have, then there is the sporophyll, a single leaf bearing the spores is known as a sporophyll. The sporophyll, then we have the structure of a sporangium, and a major sporangium has a spore mother cell, and spore mother cell, which is a 2-end structure, it undergoes meiosis and it forms spores, which is a haploid structure. The spores, after landing on a suitable substratum, they germinate and they form the gametophyte. So, since the gametophyte of Pteris is monoecious, it has both the archegonium and the antheridium. So, when these mature, they form the antherozoids here, ciliated antherozoids and an egg cell, and these two fertilize to form the zygote, which is again a diploid structure. Now, this zygote develops to form the young sporophyte, and gradually the gametophyte they disintegrate, and from the young sporophyte again a sporophytic plant is formed. So, this is the life cycle of Pteris species. Thank you.